Okay, so I will go through some of these key points in the slides. Then we will look at the differential variability analysis, the DV method, using example data, the data I already upload into here. So if you download, you should be able to have this prepared MAT file for you. If you want to process the data by yourself, you can use this sample accession number. You'll get very similar results. Then we will look at SVD function application in this example matrix that will be mentioned in the lecture slides. Then using this SVD sketch, this function to do a compression of image file, image matrix. Follow this example. Then we also have a code for ICA. ICA is to help you to, from a mixed signal, to work out the independent signals. Okay, so that's what I supposed to cover during the Tuesday, but Dr. Salim helped you to understand this. So the idea is start from, they're very important topic, by the way, in the machine learning or data science field. There are so many methods you can use to do this matrix decomposition or matrix factorization. So idea is like this. You know how to do matrix multiplication, then these two A and B, and get the results of the product A and B. So matrix decomposition or factorization is opposite operation. You have A and B. A and B, it's a product will be given to you, and you are not going to be gave A and B. So if you have A and B, it's very easy for you to compute the product. But now you have a product, you want to reconstruct A and B. So this is an example. This is called the Netflix example. Okay, you have user and the movie, and for the rating. So the viewers look at a movie and then give their scores from 1 to 10 or 1 to 5. So you have a very sparse matrix like this. Then you can do this decomposition to change this matrix. You consider it as a product of two matrices, and you get a user and a D component, or different type of users. And then D is another component. So D have to be matched. So if you have classify users into 10 categories, the D equals 10. And here you have 10 which different components of type of movies. Then you have movies. Okay. So you want to use this two matrices to understand how they interact between users and the movies to regenerate this response or scoring matrix. Then you replace this as a gene expression. You have samples and the genes expression. You try to decompose them into samples, then gene programs and the genes. Okay, so that's the idea behind this non-negative matrix factorization. So this is a very important operation, okay, especially in the gene expression analysis, because we have non-negative. Non-negative means anything which positive and zero. It's very useful because our expression data is bounded from 0 to a certain level. This illustration, you can see, okay, looks like there are 3 blocks. There's 1 block here, and here is, and then there's the baby tiny blocks here. So if you do the decomposition, see, into a 3, and then you have a matrix with very skinny matrix, only have 3 columns, 1, 2, 3. Okay, these are different values, and then you have those samples. Well, this multiplied by 3 rows and columns, depends on how many columns you have here, and this will give you a approximation to that if you do the product between them. The reason to do that is you try to understand 1, 2, 3, the 3 components in the original matrix. Sometimes it's obvious, like this is very obvious. You can see from the color and the distribution, there's blocks here, the 3. But if you have a matrix, like our single cell data matrix, you may not have this very clear picture. But you want to do this analytically, use numeric method to see, okay, how many components I need in order to reconstruct this, okay? If you overfit the data, you may say, okay, I need not only 3, but say 15, and you will overestimate the number of components. You get this, then you use 15 different components in order to approximate the original one. You will do a better job, but it's some kind of overfitting. So this looks simple, and you have product X and decompose into U and V, okay? But numerically, it's very challenge. So until very late in the single cell, now the data science development, people found out there's a way to quickly compute this through the iteration and updated this to weight matrices. So the operation I just show you is this optimization procedures. So that's inside the MATLAB or other languages, and you will find the equations. This operation, the computer code is almost identical to the mathematical expression here. So you can use this algorithm to do this decomposition. If you try this, multiply that, you will get this, okay? So in MATLAB, this function is called NNMF, non-negative matrix factorization. It will factor the non-negative matrix. So your input matrix A contains 0 and positive values. Then this algorithm will return 2 matrices, W and H. You need to give a K value, so you believe how many components in the system you have initial setup of this hyperparameter k. Okay, this algorithm will do the loop and try to minimize this d. Okay, the d is difference between this a and the w and h product. Okay, if you look at this, you try to make this as small as possible, right? So that's in Python. You have scalar this. You can call this function also an MF. Inside the algorithm, the same formula, okay, to do this norm, this 2 bars, and then to try to minimize this. This is a 2004 paper, and people start using this not in the single cell, but in bulk RNAs, maybe microarray data. But you have this many of samples, observables, and then those are the genes or features. So this is a general problem, and has already been used in gene expression analysis before single cell data become available. Now you just replace these samples into cells. You have this matrix of gene and the cell. And the paper is PNAS paper gave a name for these components. It's called meta-genes. Or you can consider there are multiple genes and functionally in the same group or same gene program, expression program. So in this case, the author believe this matrix contains 2 meta-genes, or there are 2 independent gene expression programs. So you can have this and that. Then you multiplicate by this matrix. So y is w and h. This is a n by 2. K equals 2, and this is a 2 by m, so the product will be n by m matrix, original. So you try to use optimization to optimize all the values in these 2 small matrices. Once you are done, the values in these 2 small matrices will give you the idea about which gene get involved in how much into the first meta-gene program or gene expression program. Which gene will contribute more or get involved more into the second meta-gene or gene expression program. So you can look at this distribution and look at the relative contribution of each genes in each gene expression program. If you have a blue, for example, this is low value, it means this particular gene, I don't know if you can see, involve more in the second gene expression program, but not in the first. And from this matrix, from H matrix, you can find the samples, whether they are more active in the first gene expression program or in the second. You can see in this, looks like the samples have been classified into 2 groups. The first 2/3 of those samples seems to be more active or be involved in the first gene expression program, while the rest of these samples are more active in the second. So you can separate their activation or profile in the meta-gene expression profile into 2 different classes. Well, there are different names. People use meta-genes nowadays. We don't use, but the same idea is components. You decompose the original matrix into, okay? You can call it gene expression program. You can call activation group, or you can name it whatever you want, but Meta-gene is the early ones. There, yeah, questions?
So meta-genes are mainly the most important genes that are responsible for the
Meta-gene is just a terminology to see, okay, it's a function group or a program. Gene get involved. It could be a sub-function of a lot of genes together. So it's not a gene itself. It's a set of genes work together towards that particular direction or function group.
like maybe for the inflammatory activity of the cell, something like
Yeah, you can replace it. Okay, the first component is inflammation, inflammatory response. And the second could be the something else, is matrix, extracellular matrix production. So those functional domain or description or program.
So, from what I understood, it is the main set of genes that are responsible for the function of that cell, something like that?
Yeah,
Okay.
yeah. You try to find, if you don't know, for example, you have this matrix, you cannot see, but once you summarize them into this, you start to see, okay, the gene involved in these programs. Mm-hmm. So some gene may be very useful and they involved in a lot of programs. So they have values in all these programs and weighted or contribute a little bit. So 30%, 30%, 40%, something like that, involving the three, give you a relative contribution to the program. Yeah. Yeah. So you will find a lot of this review paper applications. For example, this group have used this extensively to discover what we call the activation group, or knowledge gene expression program. So that's what the name I want to use. So they put this PCA, ICA, and MF. So this is the expression matrix. And this is, in this case, you see it's not a program anymore. It's become factors or components or meta-gene. So here is called factor, and factor sample, gene factor. There are different names to call them, these components. But you see, this matrix multiplicate this, become that. Same as here, okay? This matrix multiplicate this, become that. This is called a meta-gene. Here is called factors. You can also name this like factor, and gene or molecule of interest samples. Okay, so this matrix called amplitude patterns. So they're different name, but essentially they are the same. It's just multiplication of matrices become that. This decomposes into two. This is another example. I think study this tumor and this to find the they have a cell type matrix, and then they will consider different sample is mixed of different components or different ratio of different cell type. So if you do this decomposition, you will tell each sample how many different cell type in each sample. Okay, so they play with matrices, multiplication, decompose in different ways. This approach has been used in so-called deconvolution of the bulk RNA-seq. Okay, so the idea is people consider bulk RNA-seq as a mixed of a multiple cell type. So it can be, say, 30% T cell, 30% B cell, 40% of macrophage. And so this cell type will be considered as a factor. Okay, then in this case will be K equals 3. So if you don't know this, so you have a mix of 3 cell types together. So let's see. 3 cell types, so this will be, so this is W. If you do this, then you have 3 cell type. K1, K2, K3, and then you mix them up. K, wrong way to let's do something easier. So you have cell type gene. You need to you have a gene, okay? Cell type. Gene by cell type. A matrix like this. And for this, you can get from single cell study. Okay, so this matrix you can get from single cell studies. And if you believe your sample is different cell type and mixed with a proportion, okay, so this is cell type. If you consider like n cell type, then you have n, so this match. And for each cell type, you have percentage, so sample 1, sample 2. And if you believe this is a sample 1 have a 30% of cell type 1, 30% cell type 2. So you have a percentage. Then, so this is like Y. So the product will be a gene by sample. So this will be X times Y. So decomposition, the idea is you have this measured from samples and know their gene expression. So this is measured from, and you also know this. Each gene have a, for each cell type, the expression. So you have this and that, then you want to compute Y. This is a known, this is a known, and you want to know each sample, and this sample have a 30%, 30%, 40. This sample could be 10, 20%, and 70. So you know this, you can compute Y. The Y is you want to know whether different cell type have a different proportion to be in the sample. So the patient could be 30, 30, 40 for 3 cell type. But the patient, the healthy control, maybe have a different ratio of a 3 cell type. So that's early stage. So even this 2015, people start to use this because at that time we probably have this different cell type and their marker genes. So they consider when you mixed different cell types, you should be doing this decomposition to find the proportion of each sample for relative abundance of different cell type in different samples. It's become very important. For example, here you see in the diabetic samples, you always have a cell type 2, which is consistent across all the patients, have a similar pattern. Well, in the normal, then you don't see that. Okay, so this is another deconvolution method. That's another, so they're all the same type. This is an application for the deconvolution because the resolution for spatial transcriptome in the early stage is not high enough. So every single spot you sampled, there are multiple cells in there. Most likely you have a mixed population of cells. So you want to do the deconvolution for each this spot here, okay? Consider they are mixed of the different cell types, so you can work out, okay, in this corner here, this definitely only have this red cell type, which in the middle, you see their transition phase. They're 50% this, maybe 50% purple. So you can define this relative abundance of different cell type using marker genes, this matrix. Okay, the problem become decomposition, and you observed this expression, but this expression is a mix of different cell type in a different ratio combinations. You want to know the ratio. This, okay. You can make this very sophisticated because each cell may have this neighbor cells. They form a network, and this network, if you have immediate neighbors, you should have a similar level of the relativity abundance of the same type of a cell. So you make this transition very smooth if you consider the neighborhood relationship. And again, the idea is the same. You want to have metagene. You want to have a proportion of different cell type mixed under different position, in different position, influenced by their neighbors as well. So eigen decomposition, that's something you already know. You have a matrix. This matrix is a squared matrix, so you use this matrix to rotate a vector. Well, the impact of this rotation to this vector is not to change the direction, but the same vector, but it only change the vector, the magnitude, okay? So only change by a fraction, a factor, this eigenvalue. These are the examples. So you run this on that, you get 6 times of every element in this eigenvector. So eigenvalue is 6. You see this again, if you still don't know this, you are in trouble. Okay, you should know this now. Okay, by the way, what does that mean? Why this? We just rearrange the expression. So the definition of eigenvalue, eigenvector, you will see this equation all the time. But now we just move this vector from the left to the right, and that become a function, expression. So if you have A and eigenvector lambda eigenvalue times V, you just move this to this side of equation, and then you need V transpose. Okay, because you move here, basically it's divided by this V, so you put its inverse here. I believe because this is a squared matrix, so you can do that. If it's a symmetric as well, you can also do the transpose. They are the same as the inverse. So either way, so you basically you decompose the original squared matrix into this, make a factor of 3, factorize into 3 components, and then you use the information in those to understand the property of A, or do the operation on A. So that's what you will get the expression. Okay. In the real applications, we have a lot of this so-called symmetric real values in there. If they're symmetric, you can just do this decomposition. This will become a scaling factor. It's eigenvector values, and then you have this two decomposed matrix contains multiple eigenvectors. So this is called eigendecomposition, okay, applied to squared matrix. We have a lot of squared matrix, the pairwise distance matrix. So from one city to another, the distance from city A to B and B to A are the same. So they are symmetric matrix. Covariance matrix is also symmetric squared matrix. So we can use this decomposition a lot in many cases. So if you decompose A into that, you can compute this matrix into this power or exponential easily. So for example, matrix A times squared, if you decompose them first, it becomes this decomposed times this, and then this P will be cancelled out at the end. You get P times D squared. So only thing you need to compute this D squared. D contains just these eigenvalues. So basically you translate this computation of the power of the square matrix into this compute the power of these individual values. Okay, make this computation much faster. Otherwise, when the number, for example, this is only 2, when the number becomes, say, 500 higher, it's hard to compute the matrix multiplication again and again. But if you decompose them first, you only compute, so D 500 power, then it's very easy to compute. So the thing's even more useful when you compute the exponential of matrices. Otherwise, you have to expand this to some kind of number, series numbers of those to approximate this. But if you do the decomposition, then you can just compute this exponential of the eigenvalues. Yeah, this is a small video, very old, to introduce singular value decomposition, SVD, okay?
To a mathematician, a matrix is a table of numbers which describes the relationships between the components of various kinds of systems. The system might be a bridge, an electrical circuit, a nuclear power reactor, or a sociological group. Each of the components of the system, the bridge struts, the resistors and capacitors, the neutrons, or the individual people, interact with the other components. The matrix elements represent the intensities of all these interactions. Analysis of the system using matrix techniques allows one to predict the effect of individual interactions on the overall system. Oscillations may be generated. Matrix analysis can describe the frequency and magnitude of such oscillations. This bridge near Tacoma, Washington, was designed and built before the effect of such interactions could be completely analyzed. The wind has reinforced some undesirable oscillations with unfortunate results. Modern computers are particularly well suited to solving problems involving large matrices. The designer of a system can obtain accurate predictions about the reaction of his system to external forces. The computer is used to simplify the matrix so that most of the entries will be zeros. One method is known as the singular value decomposition. The original matrix is represented as the product of three matrices. In the second of these matrices, the only non-zero elements lie on the diagonal. These elements, the singular values of the matrix, determine the frequencies of oscillation of the original system. The other two matrices must preserve the properties of the original matrix, so they are orthogonal matrices. Each is the product of elementary matrices, which affect only two rows or columns. Scientists at Los Alamos Scientific Laboratory have been studying methods for the accurate and efficient calculation of the singular value decomposition and of describing to computer users how the computation is carried out. In this three-dimensional representation of a small matrix, each pyramid depicts the magnitude of a single matrix element. The diagonal of the matrix runs from the upper left to the lower right. Orthogonal transformations will be used to introduce zeros into the matrix. The initial transformations put zeros into the first row and then the first column. Each transformation affects two other columns or rows shown here in blue. Later transformations introduce zeros into later rows and columns, while preserving the zeros already obtained. It is not possible to annihilate all the off-diagonal elements in a column, so this first stage produces a matrix with non-zero elements directly below the diagonal, as well as those on the diagonal. This film is produced by exposing a single frame each time a matrix element is altered, to illustrate roughly the computer time actually required for various phases of the computation. The elements shown in red are involved in auxiliary calculations which require time, but which do not directly alter any elements. As more zeros are introduced, the rows and columns become shorter, and each transformation requires less time. The second stage of the calculation is a repetitive or iterative process which reduces the size of the remaining off-diagonal elements, but does not zero them in a single step. During the process, new elements, shown in red, are temporarily introduced into the matrix. The calculations now being shown are primarily intended to reduce the size of the off-diagonal element at the lower right-hand corner. Once an off-diagonal element has reached a negligible magnitude, it is set to zero. The computation of the corresponding diagonal element is then complete, and the resulting singular value is shown in blue. During these iterations, Most of the other off-diagonal elements are also being reduced in size, although not as rapidly. A few of them will occasionally increase in size. This makes theoretical analysis of the method difficult, but does not seriously impair its overall effectiveness. Since elements are being depicted according to logarithmic scale, the off-diagonal elements we see are actually quite small by this time. The transformations are still changing the diagonal elements, but the changes are not visible on this scale. Once a few of the singular values have been obtained, the off-diagonal elements have become so small that each of the remaining singular values is obtained with a single iteration. The final diagonal matrix, together with the two transforming matrices which produced it, then provide the basis for a complete analysis of the original matrix and the system which it models.
Multiplication, this is m to n, so the u will be mm, so squared matrix. This should be prepared into m times n, so this dimension. So that's why you see this two column, one columns have only 0, there's no values in there. But just to make this multiplication possible. So once you have this m by n, then you can multiply it n by n matrix. So that's what the whole thing constructed. So the idea here is to decompose this matrix into different actions, okay? So decompose it into this rotation and that rotation and the stretch effects. So A is decomposed into rotate, because this is square matrix, and scaling to the one of the dimension direction, then rotate again. So if you have this, you can do rotation back and the scaling in the inverse, okay, and then rotate back. So, so you can have this rotation and the stretching movement. Again, this is you should watch from YouTube. This has been already watched many times. So this is an example we can try in MATLAB. So let's do it here, because computer session you need to have your hand on. Let's do this before break and put this matrix into your MATLAB by typing these values. Let's call this A. So let's put this A in there, and then we do VIDI to get those components. Okay, so put this A in there. Then once you have that in there, let's try VIDI the function. Why is broken? Okay. So this function, if you put A in there, you can get three outputs. So that's what I'm going to do in MATLAB by typing the matrix in. And then we look at what that means if you do the decomposition. So, so for this example, I believe that's different frequency of words. So appeared in the several books. You have 1, 2, 3, 4, 5, 6, 7 books. And the first 4 books are from a computer science, and then this medical related book, the last 3. So you have 7 books, and then you count the frequency of these keywords. So data appeared in 1, 2, 1, 5 frequency in the computer science book, but not in the medical doctor's book. But if you look at this the last keywords, they're not appeared in any of this computer science book, but appear in some frequency in the medical book. So it creates this book and keywords frequency matrix. So now we decompose this, and this will tell you, the components will tell you the category of this book. Okay, you can see those are non-zero and this non-zero. You can use this to classify books into 2 groups. And this concept strings, and this will be each term, their contribution to this category. Okay. So I'm going to type this into MATLAB and use SVD function to get this. One. Okay, I have my information, the data matrix in there. Let's see the doc SVD is a function. You will have USV, okay? Okay, so this is what we can do in MATLAB to do this SVD analysis. You have a matrix. This matrix does not have to be squared. It can be raw data. So any data you can copy even from a spreadsheet. It contains negative values is okay. 0 or fine. Positive, okay. I don't know complex numbers. We can give it a try. But this will have decomposition will be done. And then you have U. Look at the dimension. This is 7 by 5. So the U is 7 by 7. Okay? This is a U. And the V is 5 by 5. And in the middle of this is 7 by 5, okay? Make sense? Now we are going to, you can do this directly. I don't know if I need to do this transpose of V. Let's try. So if you decompose this into 3, and then you multiply them back. So I'm not so sure I do the last need to do the transposition or not. So same as I do. So if you do this US times V, and because V was given when the output has been transposed, so I need to transpose this back. So if you look at this, it's reconstruct of your original input. Okay, if you do this minus A, okay, you get this very small number, so negative 14, so it's very small. So basically it's all 0, they are the same. To validate, you can use norm of this. This is a very small number indicate the reconstructed matrix very similar to the original. A was original, has been decomposed into 3, but now it's pushed back. Okay. Let's take a several minutes break. Okay. Okay, so now we know you can use this SVD to do the decomposition. And then if you do the multiplication, and you can reconstruct the original matrix. You may say, okay, it's not so impressed. I know you can decompose them and then do the product and reconstruct them back. And so why we need to do this? So the secret is in we can evaluate S. So S is the singular values to tell you the contribution of each component. So if you look at S, only first and second, first two components is important. The rest of them, the contribution almost zero, or look at least from this expression. For this simple example, we know there are two major components, which is from computer science and the medical documents are important. But for the complex system, so you have many important components. So all the values may be filled up here, okay? But by looking at these values, you have some idea of the relative importance of each component. So what that means is we can truncate the SUSV, these three matrices, rather than use the original 4 matrices. We only need 2 of them for each. So I will give it a try and see. For the U, I may only need the first 2 components, so I will use U2 as a simplified version of U, which only have 1 and 2 components, 1 and 2, okay? So basically, I extract these 2 columns. For the S, I also make I don't know which 2. So this would be 2. I just use 2 of them, so this becomes squared. Only this, okay? So I only use 2 components, not 0 ones. V2 is a little bit tricky because I need to count. Five. I don't know how to extract this. Let's just try this. The first 2. So now I use this truncated version, S2, V2. I hope we can make it. Yeah, is this the same as the original? Yeah, so what you can see, I use a truncated version, only two major components, and reconstruct the original matrix. Okay. So this, you also get the same very similar, okay, as this without truncation. So the difference, this is very small. U2 is only 7 by 2. Original is 7 by 7. Okay, you only need top components to reconstruct. The major information is already there. So this is useful to help you to get rid of if you have a random noise in your original data. Okay, but you learn the major components and only use these major components related matrix to reconstruct the original and some kind of denoised version of this. Any questions? No? I think the illustration here is what we just described. So this A matrix will be decomposed into MM, okay? Mn, n by n. But if you believe there are only very small fraction of the major components is important, you can do the truncation. In our case, was 7 by 7, but now becomes 7 by 2. So only top 2 is useful. And this is 2 by 2. And this is 2 by 7, whatever. Then you put them together. So use a truncated version to reconstruct original and this approximation. Okay. So this is called a low rank approximation. Another way to express that, okay, two components to reconstruct or approximate the major signals in the original. You can also write, so this is right in the matrix way. This is a matrix, m by 2, 2 by 2, 2 by n. You can write them into a vector-based production. So this is a vector times a single number, okay? Then times another vector. This will reconstruct first component reconstruction of A. This become the first layer. Then you use the second most important components to construct another matrix. Because this is the end will be a matrix. This will be another matrix. You just add another layer and then to rebuild, recreate the original matrix. That's in MATLAB, that's in Julia. This is how the SVD is computed inside this function. Okay, how this is implemented. It was originally writing in Fortran many years ago. Then had been incorporated into MATLAB. There's also uses QR decomposition to compute. Okay. There are also methods that we use, so-called indirect method, to use eigendecomposition. we use this to in order to compute SVD. So in this case, because A it's not a symmetric matrix, so we cannot use eigen decomposition to do that directly. But we do this operations. So any matrix, whatever shape, if you do this times its transpose, it becomes a squared matrix. Or you do, I think that's what this also is a squared matrix. You can try, and any matrix, if you do this, so this indirect method will do this first, then to do this eigen decomposition and make this into so V U lambda U, and make this into V lambda 2 . So to do two eigen decomposition in order to get U and V. This is to show you to compute inverse of a matrix. So you know to do the inverse matrix, if you X is here, and then when you call inverse, it gives you the inverse of matrix, but the X has to be squared, okay? But this so-called pseudo inversion is okay if you have no, it's not squared. And you can do this through this SVD. You do the decomposition first. Then you inverse this S matrix. So this only have a singular values in there. So you can do this very easily. Then you compute, reconstruct X after inversion. That will give you the inversed X. So in the example, it's like all these steps. This is using SVD to do the inversion. And to do this normal function equations that give the same answer. Okay, remember this is for the regression part. Okay, so last topic is this independent component analysis. So the idea is you have mixed information that you sampled. You believe there are two sources. Then you have to sample two different locations twice. They are mixed in different ratios. So in this microphone received music at a weight of A1 from a voice B1. So this microphone close to the music source, so A1 is larger than B1. Well, this microphone close to the speaker's voice, so B2 may be larger than A2. So they are mixed in the different fractions of a different source. Once you have these two microphone sampled signals, so you put them together, and through this ICA algorithm, you will be able to reconstruct independent the source of this signal. So that's through this process, okay? So this is an example of applications. You have EEG, different channels you collect it from a different location on your skull, and then they are mixed information from multiple sources because you sampled in different locations. Then you want to run this ICA to decompose into independent source of those. That's through this you can reconstruct this source. Yeah, so that's all, and let's go to this. I think we tried this and tried this example. Let's try SVD sketch. So sketch is what is automatically to select the top components. It's just we did this, we know top 2 is the most important. Let's try this program. Image compression, okay? What we will do is to read in matrix A, okay? So this will be a matrix prepared from an image. It's color image, but you convert that into the gray scales. This become a single layer of matrix. Let's just copy this and put into MATLAB. That's your picture, and if you look at A, it's just different grayscale and values. Everything you see on this picture, each pixel is one of these numbers. So now we are going to do the decomposition for this matrix SVD. So I believe this 57 is this corner here, okay? In the middle of this white, you have, I believe, larger values. So I move the matrix to the middle, then you should see this 254, that will be white. So that's where this picture, this region. If I move to the right corner here, I should see smaller, close to dark, smaller values at this end. Yeah, you see this only 9, 10.
So lighter color will have a larger value?
Yeah,
Okay.
yeah. So that's basically one-to-one match to the data matrix. You have this integer and this picture, okay? So we have the input matrix. Now we are going to do this so-called sketching, okay? Basically, it will decompose this into SUV, okay? But only keep major ones. So you can see we will do copy. So this will decompose this into USV and then product to recompute and rebuild this matrix. And then you will have another picture, which is from a reconstructed. So this is not original, it's reconstructed. You don't see the difference. Don't see much difference, okay? Because reconstruction is really accurate. Then you can change the resolution if you do another version, but change the cutoff. So here you have this particular ratio. Back, okay. So this cutoff is most less stringent, but here you use different cutoff. This will cause a loss of information. So you use skinny or thinner components to reconstruct the original signal. So you start to lose the information. It's not as good as before, right? But still you can tell this trees, street, cars. And if you look at the matrix, it become U2 become only 48, much smaller. So you have a way to compress the information. It's a little large, but no, you use a smaller scale to capture the major components of a picture. So you can see it was using 48 components to reconstruct something like that. If you say I only want 15, so force the algorithm to give you a very small sketching ability. So this U3 we only have 15 components. You can still reconstruct a picture, but the quality, as you can imagine, is much worse than you use major components to construct, okay? Because all the pixels you cannot calculate is very accurate as before. We can put them together. All together in the one figure. Original and high level is approximation using 288 components approximation. But this is 48 and 15, you can compare. So this is application for the image compression. But if you have your data, for example, have a lot of noise signal, so maybe you want to just learn the major ones to see, okay, major image gave you the most important signal in that picture in your data, rather than to reconstruct all the noise in there. So there are applications for denoise, but here is application to compress the original signal size. Now we can download this zipped file. We will see how the ICA work with this example. So we need to download this. This will save into our download file folder. Okay. Then we need to unzip it before we can use. So I download and then open the folder. I download the folder, and this is zipped file, so I cannot use it directly. I will do the right click to extract all. So the extract file will be saved in a new folder, okay? New folder have the same name as a zipped file, but inside you have this unzipped files. Okay, I will copy the folder directory, so because I need, I don't need this. Back to my MATLAB. I'll clean my desktop here. I will change my directory to the place where the file I just download is unzipped in there. The program we will try is to do this demo ICA. Just run this. Oh, signal process. Yeah, I don't think I can run on this machine, but if you have this installed, you got it? Yeah, just run it. Yes, right, yeah. I can try with my Excel or with my laptop. I will try with my laptop so we can assume this. Okay, so this is running. Mm-hmm. Yeah, I just run this. Oh, it was okay. So I just restart this. It's your play. Okay, yeah, so, yeah. Yeah, so I think you have to try yourself back home. I have some trouble to play this. Not running. So this basically is a mix of this people count number and the background noise. And then you can use ICA to let's see whether this is decomposed. I don't know. It's hard for me to. Maybe this one. This is better. So you have three true. That's a true signal. You have a second true signal. It's like this.
Long list of American fast foods that have successfully made the long march to Asia, from Seoul to Singapore.
And then so you have three true signals. Then they will mix into mix one.
Long list of American fast foods that have successfully made the long march to Asia, from Seoul to Singapore.
Mix two. And then mix three. So you provide these three mixed. And let algorithm to separate them. So this first output. This is second separate signal. And that's a third.
Long list of American fast foods that have successfully made the long march to Asia.
So that's just to demonstrate that you mix a signal, you provide three different mix in different ways than different components. Same components, but mixed in different proportion. Then you can finally separate them. And you can try different algorithms, and they may have a different effectiveness for the separation. So this may not separate it very well. It's okay.
Long list of American
Yeah.
fast food. Long list of American fast
So this algorithm not separated them very well. The last one.
Long list of American fast foods that have
It still have mixed background noise. Yeah. Yeah, so this is basically the idea for signal. So if you have a mixed signal from other source, different scenario play, you can think about it to use this method to do decomposition, okay? So last couple minutes we can download this data and I show you how to do this DV analysis. So I just download this. It should be in this D folder. Download the folder. Now clear. It's not, oh, still laptop. Yes, sorry about that. Yeah, so that's what I just did is to download this, clean my phone, desktop, download this. From here I can find the file I downloaded, just double click. Yeah, so it depends on your version. My current version is 2.4. Then you open that file by check the MAT file. You found in your download folder and open it. Okay, label this with batch ID. You will see one from this sample and then from that sample. This is a clear data which only have fibroblasts. So they all fibroblasts. The analysis I want to show you is this DV, okay? You know the variability analysis, try to find variable genes. And you can click this, see, okay, apply to. There's a working folder and you can there's a mouse. I just click OK and go through. And then we compare blue and the red. There's two different batch of data. Then this the selected. Depends on your version. If you use old version, you may have different behavior. So this will have a table first, then ask you whether you will do this visualization, okay? If you use older version of tools, you have a visualization first. But that's the result for analysis. Okay. So that's comparison between I remember I showed you before or in the last week, right? You found the genes which those are the gene names. That's their profile, and in the two different cells. So this table is what you want. The top ones are the most differentially variable genes. You can send this top 50 genes, the most variably expressed across two samples, to a web page. So in this web page, you can send to enrichment analysis. This top 50 genes have been sent to there, and then you can take a look, the difference between them, between two samples for those genes are involved in those biological process, and they are enriched in the function of molecular function of those. Okay. Yeah. So that's to show you. You can use DE, differential expression, all the time to compare two samples. But after that, maybe you want to consider which gene become more variable or less variable across two samples using this approach. May give you some new insights about the difference between samples. For example, I can select CCA7. This is expression profile, and then it become more variable in the orange compared to blue. That's their position. So you see this gene position. You can select different genes to look at their position. So for this TRF genes, definitely become more variably expressed in the orange sample compared to this blue sample. So that's why the position of this orange sample is here, and then the blue is there, okay? That's the different genes. Okay, so that's all for this week. Next Tuesday, we will talk about the pseudo time, okay? So we are moving into the deep water of a single cell data analysis. Pseudo time will be another very powerful direction. You can understand the differentiation or development stage of the cell using only one sample, okay? All right, any other quick question? Otherwise, see you next Tuesday.
I got lost in between because my MATLAB stopped.
Oh, yes.
So after
Yeah.
this, what should I do in the pipeline?
In the pipeline, you can do analysis,
Okay.
then DV.
Okay.
It should work. Lost data. Yeah, you have older version, so you may want to use a new version. The
Okay.
behavior is slightly better, but it can be run. Yeah.